Good afternoon, buenas tardes. Hoy nosotros estamos aquí. Today it's in English, because however we are speaking to Brazilians. And boa tarde, Cristiano.
Boa tarde, good afternoon.
However, we are talking to Brazilians, and people always say to me, Cristiano, why are they talking Portuguese? But since I'm moving here to Canada, and I'm forced to learn French because people are so restricted in language, I learned that when you are in the world, you need to find a language that is more close to everybody. And unfortunately, I don't mean to talk in English. I think I just talk in poultry English. It's like
Yeah,
like just technical English, right?
yeah.
We are trying put two of us talking about microbiota today. So thank you so much, Cristiano, to be here. It's an honor to have you here. You
Well,
are
thank you for the invitation.
you are like as a Brazilian, I'm very proud of you as a Brazilian as well because of your recognizing around the world your work that have been done a lot in gut health and microbiota, and this is why we are here today. For who are at home or in whatever you are listening to us today, you can put your questions, your comments, because we are able to see it, and then we can address to Cristiano your questions. So, Chris, can you talk a little bit about your experience, how you start to looking for or to have the willing to study more about gut health and microbiota.
Yes. Well, first of all, thank you, Elizabeth, for the invitation. It's an honor for me as well to be here talking to you about this topic. Well, my studies on the microbiome started. My interest in studying microbiome started when I was doing my master's in Brazil, because I'm a veterinarian, and because of this veterinarian background, I moved to a lab to do a master's in animal science, specifically poultry nutrition. But my professor at that time, he did some microbiota work back in the 2000, in the mid of 2000s. So I was interested in studying the microbiome and the relationship with the nutrition, how nutrition could affect the microbiome of poultry. And then I moved to the US for a PhD, first to Purdue University and then to University of Georgia. And I think was a very nice move because there are so many well-known researchers in this area at University of Georgia. So I had the possibility, I had the chance to study more in deep and do more research in that regard, always linking gut microbiome, nutrition, and the immune system, mostly how the nutrition affects the immune system, right? So their interest, I started with that, their interest, and well, I moved to Texas with my Kogut to keep looking at those aspects. And because of my vet background and my nutrition background during my PhD, I think that allowed me to move to this very well-known lab to keep doing these studies on the microbiome.
I believe that your background is very interesting one because you have like a vet, you have a nutrition background, and now immunology. So you are very complete in all these aspects. And I remember when we start to talk, when I start to study it's many years ago, before you, of course, we always talk a lot about to have, for example, very clean environment, avoid microorganisms. Every time talk about microorganisms are pathogenic, and this is changing along the year. So can you give us, especially for people that are listening to us, uh, this idea about what is commensal, what it's a good and what is a bad microorganism, because there is this concept that Clostridium is always bad and Lactobacillus is always good. Can you still use this concept, Cristiano? How is your idea about that?
Yeah, no, I don't think we can. We can until a certain point, right? Because you know that many Clostridium species are good for the bird, are good for the host, right? Like let's say Clostridium perfringens is one species in a genera that has 130 other species. So, and we know that Clostridium perfringens is bad for birds, but it's also commensal. And if we have some predisposing factors, the Clostridium perfringens will proliferate, will produce the toxins, and will cause necrotic enteritis. But many others are good, right? They produce, many of them produce the short-chain fatty acids that are beneficial to the birds. So I don't think these, of course, we need to manage in a certain way to provide good microorganisms to the birds when we place the birds in a barn. But we need to keep in mind that, and that was something that I learned during my PhD. When we had a clean litter, we had the worst necrotic enteritis outbreaks. So I believe if the litter is very well kept, like the management of the litter is well done, I think that litter will provide good microorganisms to start colonizing the bird at the moment we place those birds at the farm, right? So I think the microbiome, the first microbiome that the bird will come in contact will be responsible for colonizing. And we need to be very careful because if, let's say, Salmonella is going to colonize first, so that Salmonella will keep, and it will be very hard to get rid of the Salmonella in the gut. So we need to make sure, we need to adopt some strategies to seed the intestine. We need to provide good bacteria to start the colonization and confer that colonization resistance to the bird. So I think that concept that all of the bacteria cause disease has been changing, and it changed and is changing, right? Because you know many of them are commensal, many of them are good bacteria. Many of them produce antimicrobial peptides. They produce, as I said, short-chain fatty acids that modulate the host itself. They modulate the immune system. They modulate all of the things that we can think of: brain development, muscle development, bone development. But they also secrete the substances that is modulating the microbiome itself. Let's say, for example, we know that the microorganisms have these cross-feeding mechanism. Let's say, like lactobacillus, they produce lactate that lower the pH of the gut and have effect against acid lactic sensitive bacteria. But they can also provide, this lactate can also be used by the butyrate producing bacteria to produce butyrate. So there is this very well mechanism between microorganisms to keep the health of the gut. And once they are established, I think is of course it will happen, but is this commensal bacteria will provide this colonization resistance that to avoid or to prevent the proliferation and establishment and proliferation of some bad bacteria, such as we know, Salmonella, E. coli, Helicobacter, or Clostridium perfringens. But we need to keep in mind that many, as I said, many of those bacteria are commensal, and they will proliferate if there is any predisposing factor. In the case of Clostridium perfringens, I think coccidiosis is a very well-known predisposing factor for necrotic enteritis, right? So yeah, I think we need to change the way we are treating. We cannot get rid of the microorganisms. Mainly now in this era, in this non-antibiotic era, we need to learn how to manipulate and how to get the benefits out of these microorganisms.
So I have this is what you are saying. It's interesting because Gilson Gomes just left a questions here about. He also saying that from his experience that he also saw that the birds rising the second and third litter has also better performance than the first one. It's not just disease, but also performance that is affected by it. And you know, Cristiano, you and myself come from Universidade Federal de Paraná, and we live in the biggest producer area of broilers. I can say the word because it was really the exporter area in Brazil. We produce a lot of broilers out there, and we since we are child, we know about broilers, and we know that when you have that bad barn, that they say the bad house is always bad, and you have a good one, and good one is always good. And now I think that we can correlate it with a microbiota. So if you have like a selections of a good microbiota, you have a good growing of animals, a good performance, a good health. But also if you have, for example, a dysbiosis, some management conditions that change the balance between the microorganisms, you have it for many flocks, not just the flock that happens the problems. And what Gilson asked to us is asked to you basically. It's like in Europe and even here in Canada, the reuse of a litter is not allowed, right? So he said, what is the message that you have for these colleagues that live in Canada or in Europe, where the litter is not, the reuse of litter is not allowed? Do you think that in the future it's possible for them to review this concept? How is your opinion about that?
I think it's a waste of resources, right? Because if, let's say, the first flock didn't have any problem, disease, you can manage the litter to reuse and get the benefits of that. I think I'm not going to give any recommendation to the authorities, but I think it's a waste because,
Anyway,
as you said,
and waste of knowledge as well, because
yes, absolutely,
there's a lot of
absolutely.
knowledge behind this, right?
Of course, because as Yusun said, and when we talk to veterinarians that work in the field, like the best performances they get is in later, like second, third, or fourth cycle of the litter, not in the first one. So yes, I think it's a waste of resources, of knowledge, and I think we should learn, of course, doing a good management of that litter, allowing for the good bacteria to survive, and let's say environmental management, ventilation management, to avoid the proliferation of bad bacteria, to avoid the moisture, the high moisture of that litter, to provide good bacteria to the animals. And I think, of course, in the absence of that, we need to think of good ways or strategies to try to provide as early as possible a good bacteria to the birds, because they need to be in contact with good bacteria to colonize the gut. Maybe like using spraying the egg with probiotics or prebiotics or anything that can stimulate the growth of good bacteria in the gut. I think that should be that should be used as well to try to provide beneficial microorganisms to the birds.
Cristiano, I want to also thank you, Gilson, for the questions. And now of you that are listening to us, you are invited to let your questions there. You can write in English, you can write in Portuguese, you can write in Spanish. It's up to there. I'm not going to French. And then we can translate here for the Cristiano. Cristiano, I one point that I think that is very interesting with the new knowledge that we have about the microbiota is the relationship between we reduce the use of antibiotics. Every time that, for example, Mike Kogut present his work, his immunity, he puts about the dysbiosis, that is the imbalance between the microbiota and the host immunity, and he put as a factor that causes dysbiosis is use of antimicrobi antibiotics. So can you discuss a little bit about how these antibiotics, because in the past, sometimes people, how could this, if in the past I use a lot of antibiotic all the time, and now I'm learning that antibiotic is causing dysbiosis. Can you just explain for us a little bit how is this relationship between antibiotics and microbiota, and why we should be careful when you use it to establish a healthy microbiota?
Yeah, well, I think for a long time, the poultry industry used antibiotics as growth promoters. And so that selected a certain microbiota in the birds, which was a good bacteria, which was a good microbiota. But now with the starting removing this bacteria, these antibiotics, so other bacteria, other microorganisms started to establish, or a new microbiota started to establish. And I don't know how is the situation in Canada, but here in the US, only a few antibiotics are used as growth promoters and others are used as treatment. And I think when we think about using antibiotics as treatment, that may cause a dysbiosis. That can lead a dysbiosis for sure, because it's going, can you imagine, it's going to kill the bad bacteria, but it's going to kill the good bacteria as well, right? So that is going to cause a dysbiosis, and we don't know if there is any. As we know, there are resistant bacteria, bad bacteria that are resistant to antibiotics, and those bacteria will proliferate and will take the chance of the use of antibiotics. So I think when the industry was using antibiotics as growth promoters, it was very easy to manipulate, and the microbiota, in my opinion, was kind of well established. But now with the removal, we need to learn how to use different strategies, feed ingredients, feed additives, or different nutrients in the diet to select these good bacteria. And having in mind that if we are going to use antibiotics to treat a disease, we are going to have a dysbiosis. In humans, in rats, it shows that dysbiosis generated by antibiotics may have effect not only in the gut, but also increasing the susceptibility to other diseases, like systemic diseases, metabolic diseases. So I think we need to be very careful when we use antibiotics for treatment. Always thinking that the microbiota, the number of microorganisms will reduce, and we need to find a way to try to replace or to give those microorganisms back, the good microorganisms, the commensal microbiota, back to the birds.
I mean, some situations, like I have been many times in field, I have been professor, and one thing that happens to me when I was so young, and I think that happens with each one that start in the field, is that we know that microbiota changes with the changes of diet. For example, you are giving like start diet, then you put a grower diet, normally with higher level of fat that is different from the start one. And when you change it, you need to have these adaptations of the microbiota. And during this adaptation, sometimes you're gonna see some signs of diarrhea or feed pass through in the feces, feed in the feces as a result of this like transition period, right? So normally when you don't have so much experience or you start to work, you are so worried about what is gonna happen with the flock. And when this happens, the first things that normally a young professional think is like, oh my God, something happens. I need to start with antibiotic treatment right now. What's your opinion about that? Like if you just change the diet today, and then they start to present some diarrhea, some passage of feed tomorrow and after tomorrow, and then I visit and I saw it. What's your recommendation in this situation? Is the time is the right time to put antibiotic in that?
No, I don't think so. It's the right time because if you think the microbiota, if you are changing the feed, in my opinion, the diet, the feed is the major player to change the microbiota. If we are changing the amount of different substrates, different nutrients, as you said, fat or protein, amino acids in the diet, and that is already changing the microbiota, and we are going to see feed particles in the excreta. And if you give antibiotics, that I think I don't know what people in the field are using, if that is a good strategy, but I don't think that might be a good strategy. I think we need to provide, instead of that, we need to provide uh, good quality ingredients, uh, um, uh, very available nutrients like amino acids, minerals that can be absorbed by the bird. And try to be careful, maybe use some other more natural, let's say more natural alternatives to try to mitigate that that phase, that feeding change, the feeding change consequences that we know that can occur. Maybe that is a good target when we are talking about manipulating the microbiome. That is a good target for to test feed additives, prebiotics, probiotics, essential oils, or even organic acids. Maybe organic acids is a good alternative or an essential oils because they also have antimicrobial activity, and that can help the bird or even improving the digestive function, increase the secretion of enzymes or the activity of the enzymes in the intestine, in the brush border, and that will also, let's say, increase the digestibility and reduce the amount of feed that is reaching the lower part of the gut and leading to dysbiosis. So yeah, my opinion is that if we are already have dysbiosis due to this change in the feed, if we really had antibiotic to treat anything, I think the the, the consequences may be even higher.
Will be worse,
Yeah.
right? I think I also think that this is the time that you really are gonna see the efficiency of alternative product that you have, the additive that you have in the diet,
Yeah,
and
absolutely.
be patient because the microbiota need also to adapt that, and maybe one day later you're gonna go there and see nothing,
Yeah.
if
Yeah.
you don't treat or do anything. Another aspect that I think it's important. There is Carlos Rodriguez here. Thank you. He sent questions for us. Could you tell us, in your opinion, how is working essential oils to promote good bacteria?
I think, well, we know that some of these essential oils, they have antimicrobial activity, but as I said, they can have effect against bad bacteria, but they can also improve the digestive function or improve the secretion of, let's say, IgA, which is immunoglobulin that is present in the mucosa, in the gut, that is selecting or preventing the colonization of bad bacteria, bad pathogenic bacteria. So I think that they have different mechanisms of action, a direct effect on the microbiota, but also effect on the host, improving the digestive function, the immune response of the mucosa, of the intestinal mucosa, that will select a beneficial bacteria. And in some cases, I have seen lately some studies, let's say, effects on the Eimeria, against Eimeria. We know that we need to start looking for these alternative feed additives, and they might be good to prevent the establishment or the appearance of these predisposing factors that can lead to the development and proliferation of other microorganisms and the occurrence of these other diseases. So I think it's good. Of course, we need to know, we need to do research on that to see how they are working, but I think it might be a good alternative. Again, I don't think that will be effective to treat a disease once you have a disease, a pathogen already established, but that can help to let's say, increase the resistance or prevent the establishment of the disease, having effect on the microbiota, but also direct effect on the host.
Yes.
And I didn't mention, but also like antioxidant effect. That is a huge thing that we need to think of at the gut level, but also like systemic, right?
Yes. There is another question here, but it's from Jorge Gonzalez. He said that you said that Salmonella is the first microorganisms to colonize the intestinal tract. I don't think so, right?
No, I didn't say that. If Salmonella is the first, if we have, like, as I said, contamination, if we place the bird in a contaminated environment, or even if the hatchery is contaminated, and if it's Salmonella, it's going to colonize, if Salmonella is one of the first colonized, I think it's going to be very hard to get rid of that. But if you have good bacteria, that is why we need to promote the establishment of good bacteria first, to prevent the colonization and maybe the proliferation of Salmonella in the gut.
So he has said, would not be better to trigger the immune system with a live vaccine to prevent the colonization with wide strain, he said about Salmonella, and this way to control the microbiota? This is the questions.
I haven't seen much, and I don't have experience on that, but I have talked to people, and that might be a good strategy. I have seen some research, some presentations with a live vaccination against Salmonella, maybe to increase the resistance, but I'm not sure how it's going now and where it is right now, what people are using in the field. So I'm not sure how that vaccination could be any strategy in that regard.
Cristiano, we talk about the microbiota, and you are talking. I'm not sure about, because in fact microbiota, it's, however, we every day have a lot of new papers going on and be published. There is still a lot of concepts that should be established, because we still have a lot of things to understand. Like yesterday, I was reading an article, and they are trying to establish the concept of microbiome. because people talk about microbiota, that it's mainly bacteria, microorganisms, and microbiome, and then they try to put it as environment and with another DNA that could be around, another genetic genes that could be in the environment, like virus and prions and everything. Can you discuss a little bit your concept, because doesn't mean that is the last one because it's changing, but
Yeah.
your concept about the difference between microbiota and microbiome?
I think, yeah, that's something when we see those studies, we need to have that very well established, and we need to know to make those differences. When we talk about microbiota, we are talking about studying a population, let's say the population of bacteria or the viral microbiota, but we focus a lot on the bacteria, and we study by sequencing that piece of the DNA of the bacterial DNA to study the diversity, the composition of the microbiota. When we talk about microbiome, then we need to take in consideration all of the other microorganisms like fungi. I think fungi play a very role, a very big role in the intestine, like let's say Saccharomyces cerevisiae, which might be used as a prebiotic, producing beneficial molecules in the gut, or uh, like the providing molecules from the wall, from this yeast wall, that will be recognized by the immune system, is also modulating the immune system and the microbiome, the microbiota itself. But also we need, when we talk about microbiome, we need to think about all of the metabolites that these microorganisms are producing. And I think that is huge, and it's where we need to go to deep our knowledge on that. We need to know what the bacteria or all of the microorganisms are doing in the gut, which metabolites they are producing. We know that they might produce antimicrobial molecules, short-chain fatty acids, vitamins, but we still don't have enough knowledge on that, I guess. That's my feeling. Let's say like how they last week you had a speaker here invited uh, uh, talking about amino acids, and I think that is something big that we need to think about, because the amino acids, if they are not absorbed, they will be used by the microorganisms. Lactobacillus, for example, they are not able to synthesize their own amino acids. They need to get from the environment. Uh, or I think one of the, I believe a lot on the metabolism and the metabolites that are produced by the bacteria when they metabolize tryptophan. It's another big player, and we don't usually supplement in the diet because the diet already provide enough. But I think the metabolites that when the bacteria metabolize tryptophan and the metabolites that are generated, I think they have a huge impact on the immune system, on the host,
Behavior,
modulating the microbiota. Hmm?
behavior as well.
The behavior as well. Yes, like being a precursor of like serotonin and many hormones that affect behavior of the bird. So I think, yeah, I think we need to learn more. We need to have more knowledge on what the microorganisms are doing, the metabolites that they are producing, and the effect that those metabolites have on the host. Instead of only knowing, of course, the composition, the diversity, it is important. I'm not saying it's not important. It is important to know, but I think what they are doing is the way to go, like to further our knowledge in that regard.
Cristiano, do you think that today we have already conditions to establish, like if I go to the field right now and take samples of feces or from the intestine of the animals and analyze microbiota, do you think you have already a signature? Let's talk about signature of microorganisms that say, oh, this is good, this is healthy, or this is not good and this is not healthy. What do you think?
Uh, that's a hard question. Let's say if we take the samples and we do the sequencing to know what microorganisms are there. That might take 1 or 2 weeks to get the results, to do the sequencing in the bioinformatic work. But I think a lot of papers are coming out showing that there are some, mainly those butyrate producing bacteria, and not only lactobacillus, because all the time when we talk about the good bacteria, we talk about lactobacillus. That might be the case, but let's say like in the Zika of the birds, that is not the case. The Zika has a lot of Clostridia bacteria, and maybe other microorganisms are beneficial. I think one of the I have seen a lot these microorganisms, which it's belong to the family Lactobacillus is Fecalibacterium praunici. I think it's one. It's an anaerobic bacteria that is showing. I think it's has a good correlation with with a healthy microbiota. It's a short chain producing, mainly butyrate producing bacteria, but that's something that we can do in the lab. I don't think we already have something that we can go to the field like and knowing for sure what is there. Yeah, as we said, a signature. I think uh, that's something we need to develop as well. Try to develop these markers and to be able to go to the field and have an answer right away, instead of having to wait 2 or 3 weeks to get some result and to interpret the results.
Yeah, this is going also for the question of Raquel Burin. Thanks, Raquel, for sending us your questions. She asked you, how do you evaluate the extrapolation of what you have from the lab, like when you do a trial, and what happened in field? She said that she felt that there is a lot of variation in
Oh yeah,
microbiome experimental and field conditions, so she probably wants you to to discuss a little bit about that.
yeah. No, I think there is a lot of variation. Even if we try to minimize those variation and try to develop some methodologies or to study gut microbiota, the intestine in the lab, I think that is still, or try to mimic the gut conditions in the lab. Even with that, I think there will be variation because we don't know what is happening. Like in the lab, the conditions is very well controlled, but like in the bird, it's something totally different. I think in the lab can give us, and I think that is something that we are seeing now with this COVID, right? The results that we get in the lab, it's something, but it doesn't mean it's going to apply it in the field. It's a good indication, I guess. I'm not saying it's not valuable, the information that we get in the lab, but I think we need to be a little bit skeptical when taking any decision only with the results we see in the lab. I think the in vitro the in vitro studies do not replace the in vivo studies. It's good for of course, it's great for basic science to know the mechanism of action of some molecules, let's say, but we might see a totally different response in the field, and I think that is normal.
Hey, Christian, and do you think that I always say people in the field, they think sometimes they are very afraid to exchange numbers, samples, or something like that. But in the case of microbiota or any kind of knowledge, like I think it's not just microbiota analysis, but metaboloma or any kind of index that you use for gut health, we need to work together, like university and industry together, because it's the only way that you can have a standard, right? So what's your opinion about that? Because I think if you don't have a mutual of effort of industry, of the research centers together, it will not be able for us to have like something that Raquel just mentions, that we can really extrapolate results because we are taking it from the field. What's your opinion about that?
No, I think you're totally right. I think we need to have this kind of, as much as possible, collaboration between academia, research, labs, and in the field to be. And when we do in experiment in the lab or in a facility, an experimental facility, we try to mimic or to give conditions like very similar conditions. We use challenge models or any feed ingredient that might give another response or can change the microbiota, but it's not the same as if we go in the field and get the samples. I think the best scenario would be to have to work with experiment, have capabilities to have experimental conditions inside of a commercial farm in the field, having a very huge barn. And I know there are places where they do that already, and go there, raise the bird as they were in the conditions, in the field conditions, and get the samples from there and bring to the lab and analyze and maybe try to be to more open relationship between academia and the industry, because if we as researchers, if we are in academia and in the or research institutions, and we don't know for sure what's going on out there, like it's hard to give an answer or to try to formulate a hypothesis or think about a solution for a problem, right? And we are gonna always keep in the lab doing our research based on our conditions in the lab or in the experimental facilities that we have. But it would be much more useful if we could go, if we could have a closer relationship with the industry. That's my opinion. I think that would be valuable for sure.
So, Cristiano, we are going to the end of our conversation. Yes, it's already. It was so good. So now I let this space for you to give your final message about for all people that are listening to us. What is the message that you want to leave?
Yeah, I think as we talk about, I think the microbiome diet is the major player when we are talking about manipulating the microbiota, like the feed additives that we use. And nowadays, I think we have, and I think we are having even more chances or capabilities to study the changes in the microbiota. There are so many products out there that are able to modulate the microbiome, that we need to understand how it affects the microbiome and the host. And maybe my opinion is that I think the future is going to be to have we need to think about feeding the host, but also we need to think about feeding the microorganisms. We need to develop methodologies, and as I know Jeff has those capabilities to develop or to formulate molecules that can reach the lower parts of the gut to manipulate the microbiota that is there. Otherwise, only what the bird did not use is going to be used by the microbiota. But we need to think about feeding the microorganisms. And I have seen so many studies on that. Like yesterday, I was reading about, I think one of the biggest players on that is when we talk about vitamins. We are talking about vitamins to supply to the host, but microorganisms may have a really high requirement on vitamins to develop and to proliferate and for the good bacteria to establish mainly in the lower gut. as well. So I think we need to think about that, as I said, also in the case of the amino acids. Besides feeding the host, we need to think about feeding the microorganisms. If we grow a microorganism in the lab, in a culture media, we try to give all of the conditions to try to grow that microorganism. I think we need to think about that in animal. We need to think about feeding the microorganisms to get the answers that we want to. And I think all of the methodologies that we have available now, I think we are able to study all of the next generation sequencing methodologies and all of the other powerful methodologies. And I think at the end of the day, we want to get, as you said, we want to get a marker or as an indicator, a signature of good bacteria and a signature of good gut health.
Thank you so much, Cristiano, for your time and for all your shared of knowledge here. And for you that want to talk more with Cristiano, he will be with us on PSA. During the PSA this year will be virtual, and Jeff will have a virtual booth out there. And Cristiano will be there to day July 21 at 11 a.m. To discuss about microbiota. So Cristiano and Dr. Furcat will be together there discussing, and all of you will be invited to come to our booth and have more discussions with Cristiano about microbiota. So thank you so much, Cristiano, for your time. Thank you all that stay with us this afternoon. Thank you so much.
Well, thank you so much, Elizabeth. It's my pleasure to collaborate.
Okay. See you. Bye-bye.
Bye.